Good afternoon, dear friends. Today we will see the methods of isolation and the methods of preservation of microorganism or the bacteria. In last lecture, I also discussed the methods of preservation and also the methods of isolation. Again, we will see the same point, that is methods of isolation and also the methods of preservation. First, the isolation of microbial pure culture. In that, first, the microorganisms are generally found in, yes, it is found in air, soil and water, and also it is in a mixed population. Then even the diseased part of plant and animal contain the great number of microorganisms which differ markedly from the microorganism of other environments. To study the specific role by specific microorganism in its environment, one must isolate the same in the pure culture. There are two important steps or major steps obtaining a pure culture. In that, first, the culture has to be diluted until, yes, in that first step, the culture has been diluted until various individual microorganisms are separated far apart on a agar surface, that after incubation they form visible colonies isolated from the colonies of other microorganisms. Yes, what is first step? The culture has to be diluted until the various individual microorganisms are separated far apart on agar surface, and that after incubation they form what? That is, they form visible colonies and isolated from the colonies of other microorganisms. This is first step. And second step, an isolated colonies has to be aseptically picked off the isolation plates. Then pure versus mixed culture. It means pure culture versus mixed culture. First, pure culture originate from first bacterium strain. In that, all colonies look like a same or look the same, and mixed culture originated from many bacteria strain, and in that the colonies having different size and shape. Yes, what is difference between pure culture and mixed culture? In that, first, in pure culture it is originate from first bacterial strain. It means single strain, and in that the all colonies look like a same. And in mixed culture it is originated from many bacterial strain, and in mixed culture the colonies have different size and shape. There are different points we are taken into consideration during the inoculation. It is important. First, the inoculation loop or the inoculating loop has to be sterilized before every inoculation of colonies from a agar plate. Yes, what is this first point? That is, the inoculation loop has been sterilized before every inoculation of colonies from the agar plate. Second, every time loop is sterilized by heat, it must be cooled before inoculating the next colony. Yes, what I am saying, second point, every time the inoculating loop is sterilized by heat and it must be cooled before their inoculation in next colony. The next, there are several methods of isolation of microorganism. First, stick plate method. Second, pour plate method. Then spread plate method and serial dilution method. So these are the methods of isolation of bacteria. I am repeating the methods. First, stick plate method, then pour plate method, then next spread plate method and serial dilution method. These are the methods of isolation. Then we will see the importance or why it is important. Because first, once purified, the isolated species can then be cultivated with knowledge that only the desired microorganism is being grown. This is first point, it is the first importance. Yes, what I am saying, first, once purified, the isolated species can then be cultivated with knowledge that only the desired microorganism is being grown. Second point, a pure culture can be correctly identified for accurate studying and also testing and also the diagnosis in a clinical environment. Then next point, for testing, for experimenting with a pure culture to ensure that the same result can be achieved regardless of how many times the test is repeated. Then the pure culture spontaneous mutation rate is low, and in pure culture clone is 99.99% is identical. So these are the common methods of isolation of pure culture. It is the process of screening a pure culture by separating one type of microbe from a mixture is known as isolation. So what is isolation? It is the process of what? It is the process of screening a pure culture by separating one type of microbes from a mixture is known as a isolation. Yes, what are the methods of isolation? Stick plate method, pour plate methods, then loop dilution in pour plate method, loop dilution technique and serial dilution technique. Then third, spread plate method, then micromanipulator method and roll tube method. We will see one by one. First, stick plate method, or it is also called as streaking method. Yes, first method, that is stick plate method or stick plate technique. In this technique or in this method, the tip of fine structure wire loop called inoculating needle consists of a wooden or glass handle with a nichrome wire at the end of which is bent to from a loop is used as a transferring the microbe from a culture. It is also called as nichrome wire loop. We will see in practical what is nichrome wire loop. It is used for inoculation. Then the straight wire is similar. Then the straight wire are similar to wire loop except they do not have loop. These are used to transfer the culture in colony from solid culture. Here we can see the nichrome wire loop. Then in such cases the colonies from solid medium is streaked on the surface of nutrient agar medium in a sterile petri dish or petri plate. This technique or this method consists of following steps. First, hold the broth culture containing tube in a left hand and shake it. Second step, sterilize the wire loop. As I said earlier, each and every time the sterilization is important. Every inoculation before the sterilization important. First, sterilize the wire loop of inoculating needle or on a burner flame, that is sterilization. Then remove the cotton plug of broth culture tube by little finger of right hand. Same this we will see in practical. Then flame the mouth of the test tube immediately. Then insert the wire loop to form a thin film and replace the cotton plug. Then thin film in the loop is streaked either a zigzag manner by removing the loop backward and forward firmly. Then care should be taken that loop should not be firmly pressed against the agar surface. It means it does not touch to the agar surface. Then incubate the petri dish in incubator at required temperature. It means which temperature as you require. Then the growth of bacteria will be visible after a overnight or after their incubation period on the streaked marks. Here we can see the images. Then next, that is pour plate method. In this method the bacterial culture and liquid agar medium are mixed together. After mixing the medium containing the culture, it is poured into sterilized petri plate and allowed to solidify, and then it is incubated. Then after the incubation our colonies are formed or the colonies are appeared on the surface of medium. So what is this method? In this method both culture medias are mixed, that is bacterial culture and liquid agar medium are mixed together. Then next, after mixing the medium containing the culture, after the mixing the medium containing the culture, it is poured into sterilized petri dishes or petri plate and allowed to solidify, and then it is incubated. After the incubation colonies are formed on the surface. Then we will see the disadvantages of this method. First, The microorganisms are trapped beneath the surface of medium when it is solidifies. Hence, surface as well as subsurface colonies are developed and it is very difficult to isolate and count the subsurface colony. Yes, what is this disadvantage? First, it is very difficult. In short, we can say that it is very difficult to isolate and also count the colonies, also count the subsurface colonies. Then next, this method is tedious and also time consuming and it required a skill. What second disadvantage, it is this method is very tedious. Then it is next, that is it is time consuming method, and last, it require or this method require a special skill. Then the microorganisms are subjected to hot shock because liquid medium is maintained at 45 degrees Celsius temperature. Then this method is unsuitable for isolation of psychrophile bacteria. Then here we can see the images, spread plate method and pour plate method. In spread plate method, the sample is pipetted on surface of agar media. And what is the quantity of sample? It is 0.1 ml or less than 0.1 ml. Then sample is spread evenly over the surface of agar media by use of sterile glass spreader. Your glass spreader, it should be sterile. Then incubation and last tip, the surface colonies. After incubation, there is formation of colonies. Then pour plate method. Then sample is in that first sample is pipetted into sterile plate. Then sterile medium is added and mixed with inoculum. Then incubation, and then there is formation of surface colonies as well as subsurface colonies. Next, that is serial dilution. In this method, or this is commonly used method to obtain the pure culture of those microorganisms that have not yet been successfully cultivated on solid media and grows on in only liquid media. Just keep in mind, serial dilution, when this method is used, or this method is commonly used for what? To obtain the pure culture of those microorganisms that have not had been successfully cultivated on solid media and grows only in liquid media. Then a microorganism that predominate in a mixed culture can be isolated in a pure form by a series of dilution. Then next, the inoculum is subjected to serial dilution in a sterile liquid medium, and a large number of tubes of sterile liquid medium are incubated or are inoculated with alerts of each successive dilution. Here we can see the serial dilution. Here we can see the image. And that first, if you take out of 1 ml of this medium and mix it with 9 ml of fresh sterile liquid medium, we would then 100 microorganism in 10 ml or 10 microorganism per ml. Here we see the calculation, that is 10 microorganism per ml or 100 microorganism per ml. If we add 1 ml of the suspension to another 9 ml of fresh sterile liquid medium, each ml would now contain a single microorganism. Then if this tube shows any microbial growth, there is very high probability that this growth has resulted from an introduction of single microorganism in the medium, and it represents the pure culture of the microorganism. This is the serial dilution. Take 1 ml and it is diluted up to the 9 ml, that is your 10 ml. It means 1 ml contain that is 10 microorganism per ml, or the 100 microorganism in 10 ml. Then spread plate method. In this method, to isolate the pure colonies, then that is this method is best for to isolate the pure colonies. Then next, in this technique, the culture is not mixed with a agar medium. Instead, it is mixed with normal saline and it is serially diluted. Yes, in this technique, the culture is not mixed with agar medium, and instead it is mixed with normal saline, and then it is serially diluted. The 0.1 ml of sample was taken from diluted mixture, which is placed on surface of this agar plate, and it is spread evenly over the surface by use of spreader. That is, the spreader it should be sterile. Then incubate the plate, that is incubation, and after incubation the colonies are formed. You can see the colonies after their incubation. The colonies are formed on the surface of agar medium or agar surface. Then yes, this is the method. Dilute the bacterial sample. The 0.1 ml of sample was taken. It is sprayed on nutrient agar plate with the help of what? Glass spreader, and this spreader it should be sterile. Then incubation, and after we will see the colonies, or we can observe the colonies on the agar surface. Then advantages of this method: it is simple method. Second, in this method only surface colonies are formed. Then next, microorganisms are not exposed to higher temperature. These are the advantages. First, it is simple method. Second, in this method only surface colonies are formed. Then next, microorganisms are not exposed to higher temperature. Then next, micromanipulator method or micromanipulator. In this method, the micromanipulator have been built which permit one to pick out a single cell from a mixed culture. What this method? Yes, this method which permit one to pick out a single cell from mixed culture, and this instrument is used in conjunction with microscope to pick a single cell or particularly for bacterial cell. Then advantages of this method: that one can be reasonably sure that the culture come from single cell, and one can obtain a strain within the species. Then disadvantages of this method: the equipment is expensive, and second disadvantage, its manipulation is very tedious and it requires skilled person or skilled operator. What are the disadvantages? First, it is expensive. In short, we can say that higher cost. Then the manipulation is very tedious, or it is difficult to manipulate. And next, it requires the skilled person or skilled operator. Then next, maintenance and preservation of pure culture. Once the microorganism has been isolated in growth pure culture, it is necessary to maintain the viability and purity of the microorganism by keeping the pure culture free from contamination. Normally in laboratories, the pure culture is transferred periodically on, onto or into the fresh medium, that is subculturing, to allow the continuous growth and the viability of the microorganisms. This transfer the sample under only the sterile condition. The transfer is always subject to aseptic condition. In short, we can say that in aseptic condition, in order to avoid any type of contamination or the all type of contamination. Then we will see the objectives of preservation: to maintain the isolated pure culture for extended period, future in a viable condition, then to avoid contamination, and the third objective, to restrict genetic change. So these are the few objectives of the preservation. Then we will see the application for academic purpose, then research purpose, then fermentation industry and biotechnological field. When it is applied in academic purpose, then research purpose, in biotechnology, then and in fermentation industry. Once the microorganism has been isolated and is grown in pure culture, it becomes necessary to maintain the viability and purity of the organism by keeping in pure culture from contamination. Since the repeated subculturing it is time consuming, then it is difficult to maintain the large number of pure cultures successfully for a long period of time. Then in addition there is a risk of genetic change as well as contamination. Therefore it is now replaced by modern method do not need to frequent subculturing. This method include refrigeration, paraffin method, cryopreservation and lyophilization. These are the methods of preservation. First, refrigeration, second method, paraffin method, then cryopreservation method, and last, lyophilization method, that is freeze drying. First, methods of preservation: periodic transfer to a fresh media, that is subculturing, then storage in a sterile soil, then third method, storage at low temperature, then preservation by overlaying the cultures with mineral oil, and last, freeze drying. First method, periodic transfer into the fresh media, that is subculturing, then storage in a sterile soil. Next method, storage at low temperature. Then next, preservation by overlaying the culture with mineral oil, and last, freeze drying, that is lyophilization. We will see one by one. Periodic transfer to a fresh media. The strains can be maintained by periodically for preparing a fresh culture from a previous stock culture. Then the culture media, the storage temperature and the time interval at which the transfer are made vary species and must be ascertained beforehand. Then the temperature and the type of media has been chosen. It should support a slower rather than a rapid rate of growth, so that time interval between transfer it can be long as possible. Many of the more common heterotrophs remain viable for several weeks or months on a medium like nutrient agar. Then transfer method has a disadvantage. Next, the transfer the method as the disadvantage of failing the prevent change in the characteristics of a strain due to the development of variants and mutants. Next, advantages: it is simple method and any special apparatus is not required. Then it is easy to recover the culture. Then next method, the risk of contamination is more. Then next disadvantage, it may be possible to change in genetic and biochemical characteristics. So what are the advantages of this method? First, it is simple method. Second, any special apparatus is not required. Then next advantage, it is easy to recover the culture. And disadvantages: first, that is risk of contamination is more. And next, it may be possible to change in genetic as well as biochemical characteristics. Next, we will see the lyophilization technique. It is also called as what? That is freeze drying. In this method, this process where the water and other solvents are removed from a frozen product. Here we can see the image of freeze dryer. Freeze drying or the lyophilization is carried out by the use of instrument, that is freeze dryer. The freeze drying, it is the process where by which the water and other solvents are removed from a frozen product via sublimation. The sublimation occurs when the frozen liquid goes directly into the gaseous state without entering into the liquid. When sublimation occurs, the liquid goes directly into the gases without entering a liquid phase. That is lyophilization. For lyophilization, we can use the lyophilizer. It is the name of instrument. It is recommended by using slow rate of cooling as this will result in the formation of vertical ice, then crystal structures, thus allowing for more efficient water sublimation from a frozen product. Yes, here we can see the image. Procedure in this process the dense cell suspension is placed in small vials. It is frozen at which temperature -60 to -70 degrees Celsius, and these vials are immediately connected to what, that is high vacuum line. And the ice which is present in frozen suspension, it is evaporate or sublim under the vacuum, and this result in dehydration of bacterial cell, and the metabolic activities of the cells are stopped, and this will result in the microbes goes into dormant state and remain viable for years. Then these vials are then sealed off under a vacuum, and it is stored at 4 degrees Celsius in a refrigerator. Then advantages of this lyophilization technique. First, only minimum storage space is required. In hundreds of lyophilized culture, it can be stored in small area. It means it requires small space. Then second advantage, remain viable for more than 30 years. The next, frequent subculturing is not required. It means there is no requirement of frequent subculturing, that is subculturing. Next, maintained without contamination. It means there is no risk of contamination. Then lyophilized strain remains genetically stable. Then last, this method is employed for preservation of sera, toxins, enzymes and other biologicals. And last, the small vials, it can be sent conveniently through the mail or other microbiological laboratory when packed in special sealed mailing container. The next preservation method, that is storage in sterile soil. In that first, it is mainly applied for preservation of sporulating microorganism. It means a single spore, that is endospore within the cell. Yes, storage in sterile soil. This method is mainly applied for preservation of sporulating microorganism. For example, Fusarium penicillium, the Alternaria reserves, then Bacillus aspergillus penicillium. These are some examples of sporulating microorganism. Sporulating microorganism, it means a single spore or endospore within the cell, which is present in the cell. Then this method involves the inoculation of 1 ml of spore, 1 ml of pore suspension into the soil, or in autoclave it is twice, and incubating at room temperature for 5 to 10 days. Here we can see the image, and it is incubated at room temperature for 5 to 10 days. Then the initial growth period allows the fungus to use available moisture and gradually to become a dormant. And next, the bottles are then stored at refrigerator, and the viability of organism is found around 70 to 80 years. Then storage at low temperature. In this method, the culture medium are stored at refrigerator or in cold room when temperature is maintained at how much degree Celsius? That is, it is 4 degrees Celsius. Then another, the liquid nitrogen, it can provide the long-term preservation of culture media. In this method, we can also use the liquid nitrogen. In this method, the suspension of microbes is prepared in a medium containing protective agent. Yes, what are those protective agents? For example, glycerol or dimethyl sulfoxide. So these are the examples of protecting agent. This agent which prevent the cell damage due to the ice crystal formation. What is the role of this protective agent? It prevents the cell damage due to the ice crystal formation, and this suspension is sealed into small ampoules or vials, and then it is frozen at -150 degrees Celsius. At this temperature range, the metabolic activities of microbes are slowed down greatly and only small quantity of nutrient will be utilized. This method cannot be used for long time because toxic product get accumulated with which can kill the microorganisms 10 to 30 years without changing its characteristics. Then next, that is preservation by overlaying the culture with mineral oil or liquid paraffin. This is last method. In this method, the sterile liquid paraffin is poured over a slant culture of microbes and stored upright at room temperature, where the subculture can also be maintained by covering the agar slant by sterile mineral oil, which is stored at room temperature, that is 0 to 5 degrees Celsius. It limits the oxygen as it reduces the microorganism metabolism, then growth as well as to cell drying during preservation. The preservation period for bacteria from a genera Azobacter and Mycobacterium is from 7 to 10 years for Bacillus, 7 to 10 years for Azobacter and Mycobacterium, and for Bacillus it is 8 to 12 years. Then advantages of this method: it is simple method, then it is mainly used for anaerobic microorganism, then it is cost effective method, and last can preserve for 10 to 15 years. So this is all about the methods of isolation and also the methods of preservation, or also the techniques of isolation and also the techniques of preservation. This is the methods or techniques. I will stop here.